Okay, so we are at the top of the hour, so we'll respect the scheduling time, and I'm going to turn this over to Hong Ma, whose lab is presenting for us all today, and let Hong be the maestro organizing who's going in what order. So Hong, over to you.
Okay, this is great. Let me just get my PowerPoint up and running. Here we go. Yeah. Okay. All right. Well, thanks so much, everyone. It's a great, great time. I can just feel the enthusiasm in the air as we're opening up and the weather's getting hotter. So hopefully we don't have to do this much longer, but for the time being, here we are, here to, share a little bit of the work in our lab with you guys, and yeah, just get everyone introduced, and hopefully the people here, we can all get acquainted with them in person soon. So, so we're an engineering research group. Oopsie, let me see. Why is my, oh, here we go. And the way that we run a research program is a little bit different. It's not quite like a life science research group that's organized around a specific problem or specific biological system, or even a set of techniques. So the way that we run our research group is that we look at biological research in clinical medicine. We ask ourselves, now, what are some of the technological challenges that are out there right now? What's preventing, what are the bottlenecks in research, in gaining knowledge? And could we develop solutions or tools to overcome these challenges? So we're really tool developers, and we're in some ways a bit disease agnostic. But of course, there are specific systems that we work with. And we do rely on some general set of advances in engineering that we try to bring over from engineering to the biological side. So I'll just briefly mention them here. So one is microfabrication. So this is the idea to make structures that sort of micrometer length scale structures. And that's very compatible with biology, because biological cells are sort of in the 10 micron range. So there's a whole suite of tools, and the way that you make things at these length scales is different than the way that you make macro things, because you can't physically handle anything, so everything has to be like monolithic. Often it's you start with a pattern, something that's you pattern with light, and then there are ways to transfer microstructures. So here I have on here, this is an example. We can create these sort of hydrogel microstructures on top of imaging wall plates. So there's a way that we can segregate cells into separated compartments. Okay, so you'll hear some of that later. Then there's microfluidics. This is the idea that you can combine microstructures with fluid flow to create sort of these structures that interact with biological systems. And again, we utilize the ability to create sort of micrometer size structures. So we can sort of replicate some of the biological systems. We could manipulate them in a way that's somewhat similar to physiology, and get different kinds of information. And then microscopy, of course, that goes without saying. There's lots of different ways to look at biological systems. But what's really neat in the last few years is this whole explosion of data analytics and AI. So there's just so much you could do based on the ability to look at images and recognize certain features. Okay, and when we sort of combine all these techniques together, there's the underlying thing, which is that with the availability of robotics and automation and computation, we can automate a lot of things. So now if you figure out how to do something once, then there is often a path to then to do that many times and in an automated way. Okay, so that's sort of a general, maybe a philosophical introduction here. So today there'll be 3 people presenting from my group, and we usually try to rotate whoever is presenting in the topics. So Erik will talk to you about what we're trying to see if we can look at red blood cells and tell you how deformable it is just from looking. Okay, Pan is going to talk to you about our study in NETosis and potential paracrine signaling in NETosis. And then Ryan is going to talk to you about his work in in ways to find rare sperm for in vitro fertilization. So, okay, I don't want to take up any more time. So why don't, Erik, do you want to get started?
Yeah, sounds good. All right, thanks for that introduction, Dr. Ma. As you heard, my name is Erik, and today I'll be presenting about some recent work we've done in the lab aimed at evaluating red blood cell deformability. So why do we want to evaluate red blood cell deformability? The primary function of red blood cells is to enable respiration in tissues by providing oxygen and removing carbon dioxide via gas exchange in the lungs. Red blood cells circumnavigate the body through arteries, veins, and the microvasculature roughly every 60 seconds, and they travel over 500 kilometers during their typical 120-day lifespan. During circulation, red blood cells repeatedly deform or change their shape to squeeze through the microvasculature, often squeezing through constrictions much narrower than the size of the unstressed cell. Therefore, the ability for the red blood cell to squeeze through small constrictions dictates its ability to facilitate gas exchange. Having a method to evaluate this deformability aids our ability to evaluate a variety of blood disorders, such as malaria or sickle cell disease, which are related to red blood cell rigidification, or evaluate the quality of a blood donation for transfusion. So we define red blood cell deformability as a cell's ability to squeeze through the microvasculature, and it is an indication of cellular age. As such, it's a biomarker for transfusion quality. More deformable red blood cells increase skin blood flow and hemoglobin levels, whereas sufficiently rigid or sufficiently aged cells get caught in the microvasculature and are targeted for phagocytic removal. Transfusion red blood cell deformability is dependent on both the donor's characteristics, such as their age, sex, diet, genetics, or activity level, and the donation storage time. The effective storage duration on red blood cell deformability is also donor dependent. Some donors' blood stores better than others. So more deformable blood is hypothesized to last longer in the body after transfusion. Therefore, there's a need to identify donors with long-lasting transfusion blood to reduce the frequency of transfusions in chronic transfusion patients. More rigid blood donations should be allocated to acute transfusion patients, such as those suffering from acute trauma injuries, as these patients just need a blood top-up and can otherwise produce an adequate number of red blood cells. So here are the main research areas of interest. We have done some recent research evaluating deformability changes during storage. These deformability changes are donor-specific. Some donors' red cells store better than others. We also aim to predict transfusion lifetime via deformability. It's hypothesized that more deformable blood will last longer in the body. And by predicting transfusion lifetime via deformability, blood donations can be allocated based on recipient need. Deformability sorting could facilitate a method for distributing donations with specific predicted longevity for acute or chronic transfusion patients. And lastly, we aim to improve the speed and accessibility of determining red blood cell deformability. There have been recent advances in computer methods, specifically in machine learning and deep learning methods for classifying microscope images. So as a bit of background, red blood cell deformability regulates the cell's rheology, or its ability to flow or act as a soft solid, its longevity, and the efficacy of its oxygen transport. Red blood cell deformability is affected by cellular dehydration, cytoskeletal integrity, cellular metabolism, and the integrity of hemoglobin. And measuring red blood cell deformability is challenging since there are a variety of techniques with no gold standard, and current techniques mainly require specialized equipment and personnel. The image shown here on the right is an example of a red blood cell squeezing through a microfluidic channel that is narrower than the height of the cell, which is a similar example to how we measure red blood cell deformability in our lab. So here I'll move on to describing the methods for microfluidic assessment of red blood cell deformability. Once a blood donation is acquired, it is processed to obtain a 1% hematocrit sample for infusion into the microfluidic ratchet sorting device, shown here. This image shows red blood cells being sorted through the constriction matrix of the microfluidic device. So sample pressure pushes the cells across the matrix, and oscillatory up and down pressure forces the cells through progressively narrower tapered constrictions and declogs any cells stuck in the constrictions. More deformable cells are sorted further up the matrix to lower numbered outlets, while more rigid cells are sorted to higher numbered outlets. This sorting produces a normal distribution of red blood cells and deformability outlets. This sorting is donor specific, illustrated by the deformability differences between the orange and green donors. So it's difficult to quantify these donor deformability differences using the normal distribution, so the information is translated to a cumulative distribution plot. The interpolated outlet number at the 50% crossover frequency is defined as the donor's rigidity score. So now moving forward to some recent results, here I'll discuss donor deformability differences tested on the day of blood bag manufacture. So this is recent work completed by Amel Islamzada, a PhD candidate in our lab. She evaluated red blood cell deformability in 14 donors, 8 of whom were male and 6 were female. Here are the cumulative distribution plots for the 14 donors. The rigidity scores depicted on the plot are the means plus or minus the standard deviations. We can see the rigidity scores range from roughly 2.5 to 4, with male donors having more deformable cells with less variation between the different donors. And now here's the same information translated to a dot plot. We can see that the most deformable female donor is similar to the most deformable male, and there were female donors who have significantly more rigid red samples than males. So now I'll discuss donor deformability differences during blood bag storage. This is also recent work completed by Amel. For these 14 donors, deformability tests were completed at regular intervals for 8 weeks. From these plots, we can clearly see that some donors' blood stores better than others. For instance, donors 2 and donor 9 have very little variance in their deformability aging curves, while donors 7 and 8 rigidify more substantially and have a wider variance in their deformability scores. Further, Amel looked at the sex-based differences in deformability.
Erik, you're muted at the moment.
Okay, I'm unmuted now. Thanks. Okay, so further, Amel looked at the sex-based differences in deformability degradation during storage. This plot shows the change in rigidity score between weeks 0 to 6 and weeks 6 to 8. It is clear that the red cell rigidification occurs at different rates for donors, with donations from males generally experiencing greater rigidification. This dot plot displays the rigidification over time in a slightly different way. It's clear that there's substantial rigidification between weeks 6 and weeks 8, comparable with the changes seen between week 0 and week 6, especially in male donors. So now I'll move on to some recent work that I completed on using computers to predict red blood cell deformability from microscopy images. So here I'll describe my general approach. A red blood cell sample from a donor is processed and infused into the microfluidic device at 1% hematocrit. The cells are sorted by their deformability levels and the donor rigidity score is determined. After sorting, the cells are removed from the device and placed in imaging well plates. Using a Nikon microscope at 10x magnification, bright field full well image scans are obtained. These full well image scans are passed through a custom computer vision segmentation algorithm, and the images are augmented by rotation to produce balanced classes of different deformability levels to train the machine learning model and to test it. And the testing and training models are separate with cells, no cells overlapping between the data sets. This training data set is used to train a convolutional neural network or a deep learning network, which learns relevant image features to detect the deformability of red blood cells applied to new and previously unseen images. Finally, the deep learning method derives a deformability score or rigidity score and this can be compared to the microfluidic method. So this is going to be a little look under the hood. What does a machine learning model actually learn? So oftentimes, machine learning models are described as black boxes. For instance, information is inputted to them, and a bunch of complicated mathematical functions are applied to the data to learn these relevant features. The model spits out a prediction, all the while the user does not see what these mathematical functions are actually doing. One worry of using machine learning tools is that the model could be learning on irrelevant confounders and not on relevant pieces of information. To confirm that we are learning relevant features in the images, we applied saliency maps to the inputted images. Saliency maps display which pixels in an image are relevant for the machine learning model to classify an image. Warmer colors indicate greater contribution to classification, and cooler colors have lesser influence. It is clear from these saliency maps, the model learns based on cell morphological features in the images, suggesting our model is successfully capturing and learning on relevant information. So this deep learning method was applied to 10 different donors with an even sex split and with a variety of blood types. The training data for all 10 donors converged, and testing accuracies ranged from 68% to 98%, with an aggregate average of 84%. Similar to Amel's work, the deformability curves and rigidity scores varied between the donors. As seen from these plots, the deep learning model, displayed as dashed lines in the plots, successfully predicts a donor's rigidity score using separate data from its training. The aggregate average and rigidity score difference between the microfluidic and deep learning methods was 0.13, which is similar to the natural variation we see in rigidity score based on donor variability or manufacturing variance. Further, the model was trained and tested on all donor data combined to see if the model could generalize deformability features common to all donors. Similarly, the model is able to predict this aggregate rigidity score as well. And finally, the deep learning rigidity score is plotted versus the microfluidic rigidity score. There is high convergence with R equals 0.93 between the rigidity score evaluations using the two methods. So finally, the deep learning model was tested on cells that were not sorted by the microfluidic device. Applying the model in this manner is an example of transfer learning, applying the model in a manner that is different from its training conditions. Shown here are examples from donors 3 and 4, clearly showing the predicted rigidity score applied to this new data set gives similar deformability evaluation as a microfluidic device. So where do we go from here? We have a mouse model study in the works where we look to track transfused cells using fluorescent labels. Blood draws are taken from the mouse at regular intervals post-transfusion, and the samples are put through the microfluidic device. We hypothesize that more rigid transfused cells are preferentially cleared. So over time, more deformable fluorescently labeled cells will remain relative to more rigid labeled cells. Results from the mouse study will inform us on the feasibility of using deformability as a biomarker for transfusion lifetime. This will be further investigated in a clinical study with chronic transfusion patients. Samples of transfusion blood will be taken and microfluidically sorted, and the rigidity score of the bags will be related to the time between subsequent transfusions for the recipient. This will inform us about the relationship between red cell deformability and transfusion lifetime for chronic transfusion patients. Finally, the deep learning deformability prediction method will be applied to donors while their blood is stored. We aim to evaluate how well the computer model can detect the storage-related morphological changes. If the machine learning model can be made more robust, it will greatly expand the access of red blood cell deformability evaluation to any lab or clinic with an adequate optical microscope. I would like to thank the members of the Multi-Scale Design Lab, the Center for Blood Research, and other collaborators who contributed to this work. Thank you.
Okay, great. Thanks, Erik. Thanks for that presentation. So I think we'll take questions at the end. So if you guys, if you have any questions, please write it into the chat, or just remember what your question is and what your questions are, and then we'll take them at the end. Okay, so let's have Pan go next.
Okay, just give me a second here. So, okay, hello everyone. Today, my name is Pan, and today I'm talking about my research. I used a kind of nanowells to investigate the role of paracrine signaling in the release of neutrophil extracellular traps. So we know that the cells typically communicate using signal molecules. They can secrete these signal molecules into the extracellular space. A liver cell with the right receptor in its membrane could grab that signal. And by doing this, the signal sending cell brings the chemical signal to the target cell. Then the target cell would respond based on what the signal it is. Today, we're going to focus on the extracellular signaling in neutrophils. So neutrophils are the most abundant white blood cells. Their activity represents one of the earliest responses. Okay, just give me a second. Their activity represents one of the earliest responses to infectious pathogens. So after being stimulated by external stimuli like viruses, bacteria, and fungi, they could perform their functions by engulfing microbes, releasing antimicrobials, and more importantly, the web-like structures called neutrophil extracellular traps. So we call it nets. And the whole process of the nets generation is called NETosis. Sorry. So clearly nets are good for us. It protects us from lots of threats. However, excessive net formation and impaired clearance of nets have been shown to contribute to inflammatory disorders. For example, nets can provide stimuli for thrombus formation by promoting platelet and red blood cells adhesion. Nets could also induce organ damage in autoimmune conditions. Overall, NET is a double-edged sword of immunity. We want to inhibit excessive NETosis, but we still don't know why and how that happens. Some studies have shown that the inflammatory mediators could be produced during NETosis, like the reactive oxygen species ROS and the chemoattractant interleukin-8. So IL-8 is also a well-known NETosis inducer. Since neutrophils have receptors that combine these mediators, we think the released mediators from activated neutrophils could trigger NETosis of other neutrophils. Passing signals over a short distance from one cell to another target cell is called paracrine signaling. We think that there might be a current sensing system during NETosis, and this kind of system has been proved in many species of bacteria. So this system means that when there are sufficient neutrophils, the concentration of the released mediators could reach a threshold level that will trigger the nearby neutrophils to NETose. So our goal here is to investigate the paracrine signaling that trigger and enhance NETosis. We think the neutrophils might have a mechanism like the bacteria. They probably use the current sensing system to coordinate the release of nets. And to prove that, we put up balls to divide cells into small groups. So here we use nanowells. And to further limit extracellular communication, we could also increase the viscosity of the medium or add inhibitors to decrease NETosis. The nanowells were actually fabricated using the mixture of PECDA prepolymer and a photoinitiator. In this video here, we can see that the nanostructures were photopolymerized using the UV light coming from the bottom. And PECDA is a favorable material for NETosis testing because of its biocompatibility, and low non-specific protein absorption. The size of the nanowell unit could be tuned by the scanning pattern and the scanning speed. We successfully made more than 1,600 units in a standard microwell. It enables high throughput assay of NETosis by dividing cells into thousands of small groups. Here are some details about the nanowell fabrication. We first load a small volume of PECDA prepolymer on the surface of a glass slide. Then we use a PDMS cover, a kind of transparent rubber, to make the prepolymer spread evenly. After that, we use the UV light to photopolymerize the nanowells. Next, we rinse the glass slide with ethanol to remove the uncured prepolymer. Lastly, we assembled a plate frame with the glass slide. In this picture here, we can see that unit size is about 80 times 80 micrometers, and the wall height ranges from 25 to 35 micrometers. So here I'm going to show you how I quantify NETosis. After adding the inducers, there are around 7 stages of the neutrophils. And for the neutrophils that can NETose, they will go through the first 3 stages. So at the beginning, the cells will be very healthy. And after staying with the whole yeast, you can see that they're very bright. And then the cells exhibit DNA decondensation. The fluorescent signal would be still strong because at that time, the cells still have intact membranes. But after the breakdown of the cell membrane, we can see that DNA go outside of cells and that the fluorescent signal drop dramatically. For other cases like dead cells, they still look very bright under the microscope. And here are some pictures from my results. For my study, I used DHL60 cells, an alternative to primary neutrophils in research studies. For the group cultured without any inducer, we can see that cells look very bright and they have rounded shape. But for the group cultured with the NETosis inducer for several hours, after several hours, we detected some blurry, cloudy structures below the yellow arrows here. Those are nets. And for the picture on the right, we can see some DNA fragments here. So here I'm going to tell you how I calculate the percentage of nets. So here are 2 examples. For the left picture, those are the group cultured without any inducers. Those are untreated cells. And we used iolomycin calcium salt as the NETosis inducer. We can see that most of cells have been treated as intact cells. But for the picture on the right, those cells have been treated with the iolomycin. And the yellow arrows here show you some examples of nets. And only part of them have been treated as intact cells. And we used the automatic cell counting to distinguish the intact cells based on the fluorescent intensity, the area of the highlighted object, and the circularity. So basically, I need to take 2 images. I will take the first image immediately after adding the inducer, the iolomycin. Since the cells cannot respond that fast, they still have all the features of the intact cells. So that's how I get the total number of cells. After around 4.5 hours incubation, I'll take another image. So for the second image, I will count the total number of intact cells. The difference of the counted cells in these 2 images is the total number of nets. And this number will be divided by the total number of cells. And finally, I get a percentage of nets. So here in this picture, in this graph here, we can see that the percentage of nets changed over time. After 5 hours, around 5 hours, the nets level didn't change that much. And the maximum value is around the 93%. And for our experiments, we chose the 4.5 hours as the incubation time because it should be long enough for the most cells to show responses and before the percentage level hits to the maximum value. To see whether the nanowells could inhibit NETosis, we conducted experiments on the regular plate and the nanowell. We added the same concentration of inducers for these two groups. And here are some results. So we can see that when the density of cells is relatively high, the NET level is lower in the nanowell group in comparison with the regular plate group. But when the density of cells becomes lower, there is a much difference between two groups. And here are some results of the NET level at different cell densities. For the untreated cells, the cells cultured without any inducer, the NET level stayed at pretty low value. But for the treated cells, we can see that a higher density of cells, the higher level of NETs. And it also shows a strong linear correlation between the cell density and the percentage of NETs. So for the future directions, we could try things like increase the medium viscosity to limit diffusion of cell signaling. So we could try the methylus, which has no biological effects. It only increased the viscosity of the medium. And one of our former lab member, Deng, she used the methylus as a medium additive to limit contamination between the nanowell units for the study of the single cell secretion analysis. So for her study, she used breast cancer cells and she added the labeling antibodies to measure the concentration of IL-8 secreted by the breast cancer cells. And for the figure B, we can see that the concentration of IL-8 secreted by the cell correlated with the fluorescence intensity. And the figure C and the figure D prove that the higher concentration of the methylus, the greater localization of the secreted IL-8. So another thing we could try is to disrupt the paracrine signaling in different ways to reduce NETosis. So here is an example. This is a neutrophil. It has been stimulated by the calcium ions, and it will trigger the ROS production. And the ROS could also enhance the NETosis. So we think we could add antioxidant or block the ROS production to decrease the ROS level in extracellular environment. If we could inhibit positive feedback loop of cell activation, we should be able to use this method to regulate NET release. So in conclusions, we used the nanowell arrays to study the NETosis. We showed that this process depends on the incubation time and the cell density. It suggests that the NETosis is facilitated by the paracrine signaling between neutrophils. So here I want to thank my supervisors and word support from the colleagues and those affiliations. So thank you all, and that's all.
Okay, thanks so much. That's really great. Yeah, once again, please put your questions into the chat, and then we'll try to get to all of them at the end. Okay, so next we'll have Ryan talk about looking for sperm.
Cool. Sounds good. Nice presentation, Pan. Thank you very much. Share my presentation now. Everyone can see it. All right, so hello, everyone. My name is Ryan. I'm from the multi-scale design lab supervised by Dr. Hongshen Ma and the Flanagan lab supervised by Dr. Ryan Flanagan. I hope you enjoy my presentation on deep learning-based automated sperm identification for non-obstructive azoospermia patients. So approximately 15% of couples experience infertility. Of the infertile men, 15% suffer from azoospermia, which is the most severe form of male infertility, where there are no detectable sperm in the ejaculate. Non-obstructive azoospermia, also known as NOAH, is caused by a defect in sperm production. To help these couples conceive, doctors use a protocol called in vitro fertilization. This procedure requires andrologists to extract sperm directly from a testis biopsy, a process known as microsurgical testicular sperm extraction, or microtesty. Only 50% of microtesty retrievals are successful, which are then injected into a single egg via intracytoplasmic sperm injection with the aim of fertilization to achieve a pregnancy. So this leads to the clinical problem. As the success rate of microtesty for NOAH patients is only 50%, this leaves half of the couples with no options for fertility. The primary reason for the failure is the inability to find extremely rare sperm among the millions of testicular cells. Shown in the figure, classic IVF can be done by placing the egg in an environment with lots of sperm, but that is not possible for NOAH patients due to the lack of sperm. Therefore, they use in vitro fertilization, intracytoplasmic sperm injection, also known as IVF-ICSI, which in theory only requires 1 sperm per egg. Currently, it's very possible for andrologists to miss a few sperm while they spend hours manually searching through tens of thousands of microscopy fields in order to identify and retrieve some sperm cells for use in ICSI. Therefore, the success rate of IVF-ICSI for NOAH patients is currently limited by human cognition and patience. If sperm can't be found, the IVF is unsuccessful. Our solution uses machine learning to automate a systematic process that not only saves the andrologist's time, but also provides a more robust and consistent method to find rare sperm. So just going back a bit on previous work. So there has been previous work done in this field before in automating sperm counting via microscopy images. And these approaches include traditional image analysis, as well as machine learning. In traditional image analysis, they use thresholding, edge detection, elliptical mass, color spaces, and other techniques to segment sperm. However, these techniques are typically not very robust across different cell types. And recently, deep learning was used to directly detect sperm from microscopy images. These studies identified sperm in semen where there are few relative contaminant cells, but are unable to deal with complex tissue like testis biopsies that include background cells of different shapes and sizes. So speaking a bit about the methodology, first, the imaging sample is prepared by staining purified sperm from a donor to create fluorescently labeled ground truths. So that's still part of the sperm sample. And then that is later mixed with testis cells to train the model to differentiate sperm from testis cells and debris. The sperm samples are purified using a swim-up assay, which is a method that makes use of sperm's motility to swim away from their sample into a different medium that doesn't contain the round cells, debris, and non-motile sperm to ensure only sperm are stained. Yeah, and then, so to find rare sperm among testis biopsies, we first develop an AI algorithm to detect sperm in sperm-only images as AIM1 in the image. And we use this to find the performance upper bounds, test sample preparation methods, and optimize for the best imaging magnification. We tested images acquired at 20x, 10x, and 4x magnification to determine the magnification that balances performance and imaging time, as 4x magnification is 25 times faster than 20x magnification by area. So we captured bright field and fluorescent images that are then fed into the UNet, a widely used convolutional neural network architecture in medical imaging to learn to identify sperm. Sperm instances will be identified using the watershed algorithm, which can identify and isolate overlapping sperm using local peaks. And finally, the model performance is evaluated through precision recall metrics and receiver operating characteristic curves by comparing the detected sperm locations with the ground truth. So when evaluating the model for its use case, unlike training, only bright field images are needed. So there's no requirements for staining to predict the sperm locations. Hence, no staining is required. So even if the clinic prefers like a different magnification or a different imaging environment, our protocol allows both transfer learning or training a new model using the same procedure. Although fixed sperm samples were imaged for the training set, the algorithm is capable of detecting live sperm just as well. In a clinical workflow, this model will automatically detect and stop automated imaging when it finds a sperm, alerting the andrologist and communicating its location, who will then verify the sperm and then extract it for intracytoplasmic sperm injection. So we've done all this, and then we've generated sperm-only training data sets and performed tests at different common magnifications, evaluating with our custom metric that compares the predicted and ground truth sperm locations to count true positives, false positives, and false negatives. Our model achieves 91% precision and 96% recall in finding sperm in microscopy bright field sperm-only images, where precision is the proportion of detected sperm that are actually sperm, and recall is the proportion of all sperm that were identified correctly. The figure demonstrates the difference in image quality and corresponding performance. The performance falls slightly at 10x magnification and then sharply drops at 4x magnification. We continue the study at 10x magnification for its imaging speed and reasonable performance. These results are an upper bound in performance for the later tests involving testis cells, as finding sperm in a sperm-only sample is much easier than an environment with many other miscellaneous cells. So next, we train the model to find sperm among testis cells, and we were able to find 74% of all sperm with 83% precision during testing. The lower performance compared to sperm-only test is expected because the model needs to differentiate the sperm cell from its other cells and debris around it, as well as varying lighting and focus conditions depending on the location in the well. So a range of cell ratios that were used depending on the well ranges from 3% to around 25% are sperm cells. And here's an example of the full detection process, beginning from a bright field image to identified locations of sperm labeled on the image. So first, labeled image A, shown on the top left, is put through the model to output a probability distribution for the pixels shown in image C, which is then thresholded for a binary image with yellow pixels being sperm in image D. Then the watershed algorithm shown in image E isolates sperm instances that are then visualized with an enclosing circle and are numbered in image F. Image B is the ground truth used to verify the predictions afterwards and is also used for training in the training sets. One interesting observation is that the model understands and looks for the tail as a large indicator of whether or not a cell is sperm, as you can see some semblance of a tail in the predictions under image C. At this stage, we optimize all the hyperparameters to maximize performance by testing different parameters. And now I'll show you a quick video, quick demo of our system running on our microscopy software. So the current sperm count is on the left, and the green circles are where the system has predicted the sperm to be after going through the steps I just described in the previous slide. The demo is intentionally slowed down so they can see the process. It will be much quicker during live operation. One main difference is that the input size of the bright field image is not the same as the patch that was used to be input into the model. So it needs to be sliced into a grid of 256 by 256 patches before the processing. In a clinical workflow, rather than the count, automated imaging will stop once a single rare sperm is found for the andrologist to verify. And then, so finally, we validated our system using rare sperm datasets. So unlike previous datasets, the sperm concentration is much lower. As you can see in the fluorescent labels, the number of training label sperm in a single image is a few hundred, while rare sperm samples could have a few or none among the hundreds of testis cells and debris. Additionally, the area near the well edges were removed from the test set to better resemble clinical imaging quality and sperm concentrations seen by andrologists, hence the improvement in performance, finding 84% of rare sperm with 86% precision. The wells used for validation contain sperm ranging from a small fraction of a percent to approximately 1%. Finally, after some more fine-tuning on the rare sperm datasets, we'll evaluate our algorithm on NOAA samples and disassociated M-testis tissue to see if we can find rare sperm in samples that are either not supposed to have sperm or have been checked to not contain sperm. Afterwards, the tests are followed by possible clinical implementations under direct guidance from andrologists. Thank you for watching my presentation. I hope you found it interesting. I'm open for questions as well as Erik and Pan. Thank you very much.